Perfect.
Okay.
That looks like it's good. All right, thank you. Okay, so the second half here, we're just gonna go over some cyanobacteria, which it's a very different approach than the marine have. So the way we deal with cyanobacteria in New Hampshire is it's sort of like a fire drill of people calling, complaining, and we respond. So where you guys are going out routinely collecting, you know, a very specific amount of water through a net, we're grabbing samples or receiving samples from whoever wants to just come and bring us stuff. So it's hard to it's always hard to track blooms, but when you see one, you know what it is, you know? So when we have samples, they usually represent maybe like the worst observed area at a beach, or they represent like a range of conditions where we might go to one side of the lake and it's totally clear and it just happens to be piling up on a beach. And this is also a little bit different than what you do with VLAP, where you're collecting, you know, phytoplankton or plankton toes and identifying everything that was at the deep site or otherwise from a plankton net. So these are whole water samples or grab samples that represent a bloom condition already. So instead of like predicting when a bloom's happening, we're responding to something that already is happening. And so the estimate of cell count is kind of rough because the concentration can like change in an instant. It can pile up to, you know, millions of times higher than it was out in the deep site. So it's kind of just representing like the conditions at that moment, which is really hard because we're usually trying to share photos to help people like look at what cyanobacteria blooms look like, but what does that mean for cell concentration, toxicity? That's very difficult to track in real time. So what we'll see is, you know, just a whole huge range of different types of conditions for blooms. They don't always just look like a blue-green cloud at the beach. Sometimes people notice that their water is all of a sudden starting to look like a slightly green hue. Sometimes people see like a black mat of something, or the water will look like someone spilled detergent in it, or there'll be flecks or flakes. So, you know, while we're monitoring a lot of water bodies routinely, we're actually, for the cyanobacteria blooms, we're responding to the strange things that people see in the water. And they don't always form like these great surface scums that pile up on beaches. Sometimes people will call and say, well, we have this other type, like Gliotrichia is a very common cyanobacteria that we see in Lake Winnipesaukee, especially around Labor Day. And they can sometimes create like a very thick green scum, but most often you see them as like these little brown orbs kind of floating around in the water. And that picture on the bottom right is actually a bloom, but you can't really see it based on like a photo from this view. So what's good about Gliotrichia is that this is cyanobacteria. They're very large. You don't really even need to bring them under the microscope to identify them. Once you start to become more familiar with what these things look like, you can just pick up a scoop of water, like, you know, grab a scoop of water and look through a clear glass and say, yeah, that's Gliotrichia. And you can count each individual colony. In this case, what's different than what Brooke mentioned in cell count, I'm counting colonies, units, filaments, and then converting that into cells, because each of these colonies here contain roughly 5,000 cells. So you're really just counting one unit. You can just say I'm just counting units, which is really the way you should do it when you're counting relative abundance and you're counting non-cyanobacteria. But when we're talking about cells or cells per milliliter, we're actually counting each of the individual cells that are in the colony. And so that can take a lot of time. So we've come up with sort of a conversion factor for each type of cyanobacteria that we see. So again, Gliotrichia is the largest, but with cyanobacteria, size and species don't really always predict toxicity. So Gliotrichia is known to produce toxins, but relative to its size, it's a low toxin producer for certain types. So We also have some other strange ones that you don't see a lot that will not create like this blue-green surface scum. Nostoc, for example, are these like blue-green marbles that you might see at the bottom of the lake. Sometimes you won't see them because, again, they're at the bottom. They don't usually float up as like a surface scum. But if you're ever at Millen Pond, they're having nostoc kind of growing in abundance in a certain part along their shoreline. And these don't really present a danger for just contact, but if you were to consume these, they do produce toxins. They're known to produce a neurotoxin that's related to causing neurodegenerative disease. And nostoc is also a delicacy in some countries, so they actually go and seek them out and consume them. So, you know, the toxicity of cyanobacteria is really strange because they are used as supplements or delicacies, and often, you know, there's no health repercussions. So it's an interesting type. But if you were to crack one of these open, you'd see all these cells inside.
Are they all toxic?
So yeah, I'll just go off on a tangent real quick on toxicity because I don't know that I really cover it in this. No, so I'll just say, like, you know, we don't try to identify a species all the time because a species here might be toxic here and it's not toxic in Europe, and it might be identified as the same species. And sometimes it has to do with the chemistry of the water. So there's been like some correlation with like iron that can turn on the genetic signal of certain toxins, and then it expresses toxicity. And there could be like all these colonies, one colony could be toxic and the other one might not be. So even though we're trying to count abundance, it doesn't always predict toxicity, and we're talking about multiple different toxins. So just like you guys in the marine hab world, they're finding new compounds all the time that are potentially toxic, or that it's really toxic in this one species in just this region of the world, and it might be a totally different thing here. And the taxonomy is rough because they're changing names all the time. Or like at NOMS the other day, they were saying that they thought instead of separating these three genera, they think that they should put them all together as the ADA complex. I don't know if I told you, sir. So Anabina used to be called Anabina, now it's Delicospermum, and then there's Aphanaesomenin, and they're all like these chains. And so this one person, through genetic studies, found that there's an ADA complex that is actually Anabina, Delicospermum, and Aphanaesomenin that are more genetically similar when you look at like 10 or 15 species. So it's so all over the place that all of the advisories that we issue are just preemptive advisories, because we can never say, oh, it's this toxic all the time, because it can change, and then the conditions at the beach can change. So, you know, our advisory process is really based on like your visual capacity to see it, and we'll tell people, if it's at the beach, like just avoid it at the beach, but maybe at your house it's fine, you know, and you have to kind of look for it, although it can be there when you can't see it. So that's sort of what scares people, is that there's a bloom on the lake. It probably is more dangerous where you can see it, but people are still worried that it's in the water at all. And the wind can blow, and then it's like, yeah, now you have a really toxic situation where maybe you didn't on the other side of the lake. So with nostoc, like if you look inside of these balls, you'll see all these tiny kinks, and they kind of look like Anabina or Delicospermum. Each of the little cells within that ball produce toxins, and they can produce them unequally, or they can produce more than one type of toxin, and then they can shut them off whenever they feel like it. So I always cringe when I hear that there's advisories based on toxicity, because I think that it's just such a dynamic process that it's better just to try to avoid it. And, you know, we're not really likely to go up to something like this and consume it, but, you know, dogs definitely are, and cyanobacteria produce taste and odor compounds that animals are attracted to. So the danger is really for like small kids and dogs, but, you know, again, the consumption of it is a concern. And nostoc can also be symbiotically within the roots or well, multiple parts of plants, but have been found to be symbiotic in the cycad tree and has been able to transfer the toxins that they produce and biomagnify up through to like the fruit of the cycad tree. So there's also the element of biomagnification through the food web for the toxins. So they don't always look like blue-green scums though, and that's sort of what we're finding in New Hampshire, is that we're finding new things all the time because we're getting more reports. And so another really common one in Lake Winnipesaukee, or we've seen in Spofford Lake as well, some of our biggest, deepest, clean water lakes really have these dark benthic mats of cyanobacteria that we don't know like how long they've been growing down there, but usually by the end of the summer, after all of the activity on the lake, these benthic mats have been dislodged from the bottom. They come up and then like wind action brings them to the beach. And the water around this stuff might not be toxic, but if you were to consume this mat, you would get a high dose of toxin very quickly because of just the amount of material that's like crammed into these mats. And one type that we're seeing is Stygonema, which does not have much research, out there on how toxic it may be. But a lot of times these mats have more than one type of cyanobacteria in them. And so it's not always homogeneously, distributed. You might find like tufts of different types of cyanobacteria over here that are different from over here. And then they appear black because they're growing deep in the water column with very low light, and they're doing just fine. They also like to attach to rocks. So this benthic mat that was in Spofford, I identified some that looked like Lingbia, which then we sent down to a lab in Florida, and they found Lingbia, but they found it to be mostly Stygonema, this other type that's in the bottom frame there, Talipothrix and Schitonema. So there's a ton of these benthic cyanobacteria that we don't see in the surface waters, and we don't know how long they've been growing at the bottom of these lakes either. So more recently, a lot of these orange looking blooms, and this has mostly been due to Warnachinia. As far as I know, there's only really one species that we're probably seeing in Warnachinia. However, it's known to produce toxins, but as people are finding this to be more abundant, we're not sure how toxic Warnachinia might be. It actually might be a low toxin producer. Sometimes when these things are mixed with other organisms, they can create funny shapes or funny colors too. And like this orange blob at the top was a mix of Warnachinia and this green alga that looks very orange, that created like this weird amorphous shape. And like if you poked it, it would kind of flake away, but it was very waxy and a very strange anomaly of sorts. But the point is that sometimes these blooms are not just one thing, and so the color and like the shape and where they go can vary because there's other things in there with them. In this case, this Warnachinia is giving off a really strange sort of like tan color, but it can also appear green. And we're trying to learn more about this because it's been a problem in a few lakes in recent years where we haven't really observed it before. There's also these Pico Cyanobacteria, which we do not see. And I know that they are in the marine system, but they're not monitored. And they're rarely monitored in fresh water as well. And so Pico Cyanobacteria are just defined by their size. They're a category. There's picoplankton that can be eukaryotic, heterotrophic, or cyanobacteria. And we're finding that a lot of our lakes have this species or genre called Aphanichapsa. And Aphanichapsa is very common, but it can be as a single cell, less than 3 microns, or it can be a colony. And so without like special microscopy like epifluorescence, it can be really hard to see them and enumerate them if they're not in a colony. And so the point is that you really have to know what you're looking at to choose the proper way of even counting it sometimes. So seeing the Pico is really difficult without these pigments, the chlorophyll and phycocyanin fluorescence, but we can enumerate them using something called a hemocytometer, which is basically a counting chamber for blood cells. So they're kind of at that caliber of being very tiny, but we can still enumerate them. It's just hard to determine if they're cyanobacteria or not. So using the fluorescence signal can really help you figure out which one it is. We also use tools like these YSI sondes or these handheld fluorometers that can help us just kind of detect the signal that cyanobacteria give off, which is mainly the phycocyanin pigment that can be unique to cyanobacteria. So it really, again, you kind of have to choose your own adventure here on like what counting method, because you wouldn't try to jam a nostoc under the scope. You wouldn't try to count all the Pico cells under the Cedric rafter. But what we've started to realize just from our records is that the most commonly observed taxa is what we used to call Anabina, now Delicospermum. I've kept them in this category so that we can track the identification of what we called it, but these names have changed quite a bit over the years. And for example, Vorticella, we think we were identified as Coelospherium in the past. So having those notes is important for looking at trends that we might have, been calling it Anabina forever, but now it's Delicospermum. We're talking about the same thing. And these images on the left are just 6 of the more common ones I see. But that Delicospermum lemmermanii is by far the most common that we see, and it's really common in June, but then they also persist all summer. And they're known to produce an anatoxin, which is a nerve toxin that is not federally regulated. That none of these toxins are. There's health advisory levels, but there's not even a health advisory level for anatoxin, which I think is probably the most concerning because it's a fast-acting nervous system toxin, so it can cause instantaneous death in dogs, but we're not putting a health advisory level on it. So again, I kind of get frustrated with toxicity here because there's so many different toxins that behave differently. There's no guidelines on them really, and the health advisories vary by country and state. But Delicospermum lemmermanii, again, is the most common under dark field. It kind of has, you can see it's like kind of yellow from the dark field view, but under just regular compound light, these are very dark coiled filaments that kind of bunch together that can also appear kind of separated too. So this category of Delicospermum itself can look very different. The top left one is called Planktothrix, which used to be called Oscillatoria. So that one is a very deep layer bloom that we see sometimes pop up in these flakes. The top right is Microcystis. That tends to be the more toxic, especially for microcystin. Then we have Vorticella, Chrysosporum, Aphanaesomenin, and Delicospermum. So I'll go through each of these, but these are kind of like the major ones that we've been seeing in recent years. So Lock Lake, so looking at the bloom, you can see there's patches, there's swirls, maybe on the other side of the lake, it's clear. But here at this beach, we're trying to identify what is kind of accumulating right here. None of this stuff really grew here. It piled up and ended here as I am here right now. If I came an hour later, it might have been clear at this beach and like not clear down the way. So it is hard to really chase these blooms around. So having a photo that you can kind of relate to the speciation, with the composition of what's in that bloom, and then having a sample that you enumerate, identify, that you can relate to toxicity is probably the most useful information out of this. But really what we're talking about when we're talking about advisories is if you see this, look out and stay away from it. We don't even have to count this to know that this is a potential danger for you and your pets. This bloom mostly contained Microcystis, Vorticella. but also had the Dolichospermum. And the thickest areas here represented over 50 million cells. So as a unit would represent a much smaller number, but because we're magnifying like how many cells per milliliter, it's like all of the little cells that are in each of these colonies that you can see, which I'll get a little bit closer here. So basically what we get is calls and complaints. So samples are collected either as a grab. We also get integrated samples using an IT tube, maybe a discrete sample that someone just identified something deeper in the water. And we have our plankton net samples as well. So knowing like how the sample is collected is important, just so you can enumerate what you have here. We identify, enumerate based on what we see, and then we freeze for the cyanotoxin analyses. We subsample, and we can only really test for a few of the toxins. Using ELISA methods, which is just a pretty basic assay, but using like HPLC, you could really get down to like all of the different types, which would be ideal, but not always practical. We issue advisories when these cell counts exceed 70,000, but like I just said, there's so many different types of cells, so one cell doesn't always equal the same as another cell. So 70,000 itself is kind of a difficult level to equate to all blooms, but we do. And the World Health Organization has suggested that 100,000 cells per milliliter is where you're getting into a higher risk situation. So I think the 70,000 actually was derived from the World Health Organization, and then the Massachusetts Department of Health took that and made it a little more conservative and based it on a toxic species. So overall, like, we're being very cautious when we see blooms because they're not always going to be toxic and they're not always equally dangerous. But nonetheless, you cannot just look at a bloom and tell how toxic it is. And so the towns are notified, signs are posted, we'll issue a press release, it'll be on our map and social media, but we will never close a beach. We'll never say, you know, you can't consume the fish here. Although you should know that you should not consume freshwater shellfish because they probably all have a load of toxins, not only cyanotoxins, but, you know, everything. So there probably should be an advisory on freshwater consumption anyways, but we just issue advisories. The other thing that's a little bit different is I'm looking at live samples, so I think that it is a lot easier. And yes, I've looked at, you know, Sarah's got plankton net toes that have much more diversity of species, but then the preserved specimen can look like off color, have degraded a little bit, might not have that distinguishing feature. But when you're looking at live stuff, especially like phytoplankton or cyanobacteria that are not moving, it's really much easier to identify. So we have sort of the same approach. You want to, in this case, we might have a grab sample or whatever type of sample in a high density plastic amber bottle. That's mostly just to keep the light out, but the high density plastic is also important for the toxins so that they're not binding to a flimsy plastic. So you end up with a sample in a brown bottle, and the same way, you want to gently invert it. You don't want to shake it, you don't want to swirl it, you just kind of want to like gently invert it 3 times. Get your counting chamber ready, subsample 1 ml from the middle, and the same way that Brooke showed us is to prepare the Cedric rafter in that way. I scan the sample first so I can see the diversity and see what I'm getting into, but I also have to fine focus up and down because the Cedric rafter cell itself is pretty thick, and so you can miss like a whole row of organisms that are floating above. Because they're not preserved, they're not all sunk down to the bottom. So they can be like anywhere in this kind of deep chamber, basically. What I'll ask myself is, does the sample have enough units to count for accuracy? So in phytoplankton counting, you're trying to achieve a 100 to 300 unit counts for accuracy, especially with diversity of species. But with the bloom, you're already dealing with a very highly concentrated sample. So you kind of know already that you're probably going to find cyanobacteria, and if anything, you're probably going to have to dilute it. But what I'll have to do at that time is, if I determine that I need to dilute it, I might actually change the counting chamber to something else. Are the colonies like overlapping? Can you clearly identify the taxa, or do you need to get closer? Because sometimes with the Cedric rafter, like Brooke mentioned, you can't get up to the highest magnification without like breaking the slide. So sometimes I'll subsample and put it on like a flat slide just so I can really see it if I have any question. And again, are there multiple dimensions to focus on? Because this is, again, dealing with live stuff. And they're all a little bit different. So looking at a very highly concentrated sample at 40x, or the scanning objective of 4x, can just look like a whole mosaic of crazy stuff going on. But when you find focus, you can see that some of them are floating above, so you might miss a whole subset of those, or you might be on the wrong plane the whole time. So you really do need to kind of really scan around first to determine. This sample would be too concentrated to really accurately count, but there's enough there to also do like a quick count and just maybe count what's in the field of view. It kind of depends on like how busy I am that moment. But as you get closer, you can see like all of the different types of cells and all of the kinks. And each of these things that you see here are just units. They're not all of the cells. You have to count the cells within each of these units. So at the 200x view, if you look at one of these big blobs, this is a Microcystis colony that probably has like 1,000 cells in it. So I might count it and then convert it later. But you have to have an understanding of how big these things can get, and they can get really big. So sometimes I'll see a colony that's so big that I have to, like Brooke mentioned, like she had to like, you know, maybe break it down into these segments. I would count, you know, these areas, like, okay, this amorphous blob is so big, maybe I can't even count it, or I'll count that one colony as like 10. So that it's easier for me to count, because it does get really concentrated. So I do have a whole stepwise I won't get into, but basically I scan it using the Cedric rafter cell first, and then I will count per unit volume. I do find that the Palmer-Maloney counting chamber is the best for phytoplankton counting in this case. And it's very similar to the Cedric rafter, but it's only 1/10 of a mil, and you count the entire chamber. So instead of 1 mil, you're counting 1/10 of it, and you'll probably get better accuracy because it's not as deep and it's more finite for phytoplankton counting. We might also have these huge colonies that I have to kind of break down and then use the hemocytometer, just like I would for Pico. So we don't have one standard way of approaching blooms because they're all very different. And so some questions that I ask myself when I'm identifying is, does the cell have chloroplasts? You know, it might be hard to really identify if a cell has chloroplasts, but you're looking for like what cyanobacteria have and do not have. So sometimes, like if you see there's a flagella, well then it's not cyanobacteria. If it has organelles, cilia, eye spots, if it has these other distinguishing characteristics that other algae have, it's probably not cyanobacteria. What is the cell color? You will sometimes see like a whole range of cell colors for cyanobacteria, but again, you're trying to identify if there's a chloroplast within the cells, which can be really hard to see at certain views. So you're kind of have to like hone in on the cell and see, is there any granulation? Can you see like these little ribosomal like dots in there, tiny specks in the cell, or do you see like big organelles? What is the cell size? So these colonies can be really huge, but then the cell size itself can be a distinguishing factor when you go through the taxonomic references. What is the form or the shape? So I'm calling all of these units rather than cells because, again, one colony could have like thousands of cells, or a filament could have a different number of cells. And really what we're trying to get at is the biovolume of the cyanobacteria. But when we report cell count, we're talking about how many cells, not how many colonies or units. That can be hard. So I have created a cell factor conversion table, and I've compared it to Vermont's estimate. And this can vary if you have a lake that you find like has a bloom that you haven't seen the species, or maybe it's a larger colony than you're used to. You have to do the legwork to figure out what would be the average cell count per colony or unit here. And so I've come up with like a little cheat sheet on what we normally see. So it's all a pretty rough estimate. Like if Brooke and I were to duplicate sampling or counting here, a good replicate of like maybe 10,000 cells off would be good. We're talking about cells. So it's very different. So there are a lot of really great taxonomic and image-based keys out there, and there's more than this, but I just wanted to include some of the links here because I think these are good beginner kind of image-based guides to cyanobacteria. Some that we have in the lab, but all of these links can bring you to sort of these image galleries of like the more common ones you see. A really great one that I referenced, but it's Northwest, is Robin Matthews' key. And it's great because it's all image-based and it kind of guides you through what you might be seeing. And so she has this kind of broken into these categories. Okay, choose the best matching row. The cyanobacteria cells will not have chloroplasts, flagella, cilia, eye spots. They'll give examples, and so you can kind of go through and click on the images and see, like, does it look more like that or does it look like more like that, so that you can skip the difficult taxonomic references here. One that's like a little closer to home is from UNH. The Center for Freshwater Biology has like a bunch of taxonomic references that are very basic, not always correct either, but they're images that allow you to become more familiar with what we see in New Hampshire. One of them being the Dirty Dozen key is just sort of like a photo gallery, basically, of the different types that we commonly see in New England. So, if you're looking at cyanobacteria, then you can really hone in on like some of the like gallery images for comparison, because it could be just as simple as that. When you click on the Dirty Dozen key here, it has a page that you can do like a quick lookup for each of these and their known or associated toxins, and then also their reported taste and odor issues, which is also very useful for like drinking water purposes. But all of these different types are known to produce a whole suite of different toxins, and the information and data on that is also changing all the time and can really contradict what's happening in the other side of the world. But what I like about this key especially is that it gives a lot of different examples of the organism you might be looking at and a general description. So it's hard to read this from here, but this is Aphanaesomenin. It's known to produce saxitoxin, actually, very similar to marine habs. They kind of raft together, and so they can look very different depending on how big the rafts get, but individual filaments are characteristic in their cell division and how big they are, and the color and the granulation in the cell. So you do want to supplement this with a taxonomic reference, but at least the image-based approach can get you to the genre or the type that you're looking at. And so Aphanaesomenin is one of the more common ones we see, but then there's years where we don't see it at all, and a lot of times it's mixed with other types of cyanobacteria. Microcystis is a very common one, and it can look very different depending on the time of year, the species, and people are starting to say that Microcystis does not even have that many species, actually, that it just morphologically changes based on the chemistry of the water. So someone like cultured Microcystis and put it through all of these different scenarios of like more or less chloride, changing the pH, and it actually changes morphotype, so that we think that there's actually not as many species as there might be reported, and that this can just like kind of morphologically change. and can grow to like enormous sizes. So like this picture on the right is actually a colony that like I couldn't even focus on any part of it because it was so three-dimensional that you're like, okay, over here I got like it in view, but you're missing millions of cells in a single colony. And then Anabina. So now called Dolichospermum, there are still types called Anabina, and they are more associated with benthic types now, but again, the names on these change quite a bit. But I like this one because there are some very distinguishing features about it, and it is the most common. So when you're looking at Anabina or Dolichospermum cells, you're going to see they're kind of like chain beads, like a necklace, and then they're going to have a few special cells on there. One is like a vegetative cell that helps them grow, called actinides. But then more so, you'll see heterocysts, and they're going to look like these clear, sort of bluish colored, sometimes hollow looking round cells that look a little bit different than the other chain of cells in the colony. And those help the organism fix nitrogen from the atmosphere. So while cyanobacteria are not truly algae, they do behave like phytoplankton in that they utilize the sun's energy, but they're also bacteria, so they can kind of make their own nutrients, and then some of them have these specialized cells. So again, Dolichospermum is really easy to identify because of those specialized cells and how they're arranged. They're very easy to count often, but I also wonder if because they're so easy that maybe that's why they've been identified as the most common. They can also be like sort of on one plane of view where the other stuff you can't see. So it's one of those things that I don't think we have a lot of good information on the history of what blooms have really been. And in the spring, they can look very different than in the fall. By the fall, they start to like unravel. So this top one is a typical form. Here. This is Dolichospermum lemmermanii. Each of these like little coils here are beads that make one big colony. So this colony here is roughly 250 cells. But then in the fall, they kind of unravel, and then they drop their vegetative cells to the bottom. So all of these big actinides here are just ready to like rest for the winter and become a new bloom in the spring. But they can be single filaments, they can be these tightly coiled springs. There's a lot of different forms of the Dolichospermum. And again, they produce anatoxin, so it's something that we're worried about. And then there's Oscillatoria, and this has also been called Planktothrix. Other types of filamentous cyanobacteria like Lyngbia and Formidium can also be easily confused with these, and the names have changed. But we often see Oscillatoria, as the name suggests, it can oscillate and it can regulate its buoyancy, and we find it deeper in the water column. So sometimes there could be a bloom going on at like 7 meters deep. We don't even see it until it comes up to the surface in these like chunks. So just for practice, which colony is not cyanobacteria here? Sometimes it's like what does not look like the other is another way of approaching it. Could you guys guess which one is not cyanobacteria in this image? Probably not from this view, but maybe. Well, it's hard because of the color, I think, too. But this here is not cyanobacteria. This is called Bacteriococcus. It's that really orange one I showed you before that can also look green. So this one big colony is a green alga, and then all these other big darker blobs here are Microcystis, and then all of these little coils in between are Dolichospermum. So it definitely takes practice, it takes variety, repetition, sometimes a second opinion, a lot of times the second opinion. But sometimes you also just see the same thing over and over again, and you're like, I know what that is, I'm gonna count it real quick, that's it. It's when like a new species pops up, they're like, oh, great. Like, what is this? And then we have some great resources like that slide I showed you with all the links, but one of those people from Phycotech and St. Armand, you can basically like email her, upload an image and email her, and she'll get back to you pretty quickly too, which is awesome because there's not too many services like that. And also, it's just important not to get too distracted, especially with live stuff, because it's like, oh, look at that pretty, oh, wait a second, what am I doing? And yeah, and I really love zooplankton. So then I'm like chasing them around. I lost my place and where I slide. But I also want
not be doing that.
it's live stuff is really fun, but it's also, yeah, stay focused. No pun intended. But oh no. No. Some of the zooplankton though, I feel like they're so cute, yeah. But also like this Dolichospermum that we're seeing a lot of, this one species called Lemmermanni, it also will have these little rotifer type things that make it difficult. So this right here is a colony of Vorticella, and they like to stalk onto Dolichospermum and take them for a ride. And so you can just see like a big colony zoom by because you have these things like a tumbleweed going by. Yeah, so in a case like this, I will have to just like quickly like kind of come back on the scanning objective and say, yeah, I see you over there. Like that one's moving around. Where most of them are not moving, but also sometimes like these filaments, if you're really looking at them, you'll start to see them just kind of glide away, and that's common too. So my approach is a little bit different with cedric rafters. I don't do column counting, and I don't do fixed column counting. So I'll usually scan the whole cedric rafter to see the diversity, and then I will count at least 3 rows. However many I count, I have to determine what the volume was. So the grids really help in that way, but more so I think that the better accuracy for cyanobacteria, live cyanobacteria, is using the Palmer-Maloney. It's only 1/10 of a mil. It flattens it more, but you do have to count the whole chamber. And but then you have a quick conversion. And again, it's easier when you're counting units, no matter how big they are. However, you might find that you have to come up with the cell conversion yourself. So instead of using a cheat sheet, you might find like, well, I can see all of these Dolichospermum fragments are maybe used to be a big colony that represented 250, but now they're kind of like broken up into little filaments, and I can't really estimate how many there would have been in a colony. That happens with fresh samples that might be, you know, like the bloom is, how long has the bloom been there? It's viable, and then it starts to decay and die out. So these big colonies start to like put off cells and like break apart. And so you might be grabbing a sample that's already starting to break apart a bit, and you have to adjust the cell conversion factor. But in the end, it's all a big estimate, and it's you know, it's really okay if you're, like I said, like even 10,000 cells off is a pretty good replicate between 2 people, because there's just so many cells per colony, and that's the way we're doing things now. But I think in the future, we're probably going to be focused more on biovolume and the pigment signals. That's sort of the EPA's approach right now, is that like, okay, this whole cell count stuff is so difficult. Let's just use a pigment to analyze how, like, what's the relative color in the water that might be cyanobacteria. Again, in the end, like if you have 1 million cells of something that you're not sure is toxic or not, like it's more of just an advisory. So the stakes are not as high because we're not closing down a business. Well, in a way we are closing down a whole town, and people will argue that we're ruining, you know, a town's economy by even having an advisory, even without a true closure. It brings, you know, bad press for sure. But I think, again, like the danger is really for our dogs, you know, and our pets. It's just, you're not likely to go and drink water at the beach, right? But kids do, and I've seen kids drink salt water, so I wouldn't put it past them, you know? Yeah, right? Like, it's just, I don't know. So it's just more of the exposure. But I will say the exposure can be biomagnification through the food web. It can be aerosols. It can be, you know, skin irritation. So a lot of times people will, if they're swimming in it, will get eye and ear irritations, like they've had like swimmer's ear or something. So the symptoms can overlap with a lot of other stuff, like gastroenteritis is common. But that's all I had. I wanted to be quick because I was trying to keep it to a marine hab day. I didn't bring a sample to look at either, but this is just a very brief, you know, dirty way of looking at the cyanobacteria. But I have to say there's a lot of great image-based galleries, and once you start looking at them, you'll be like, oh yeah, I know you. They had a cute, you know, sort of name for the three most common, is Annie, Fanny, and Mike. So Annie, Annabina, which is now Dolichospermum. So people are like, let's call it Dolly. Fanny, Aphanaesomenin, and Mike, Microcystis. But in New Hampshire, those are probably the more common ones we see. Again, they're not always toxic all the time, and some produce more than one type of toxin. And researchers are finding that to change all the time, basically. So that's all I had. So I don't know if you guys have any questions or if you want to go back to anything, but that's basically all I have here.
If you want to stop recording, I think you can just go back to the dots, yeah. And then